Hi, my name is Valentina Ortega and I am a sophomore at American Heritage School in Plantation, Florida. This year, my project focused on finding the molecular asthmatic effects of Pichia-Kudryavtsevi yeast on A549 alveolar epithelial cells. Asthma is a multicellular respiratory disease characterized by the obstruction of the airway through mucus overproduction and smooth muscle inflammation. Currently, over 300 million people worldwide suffer from asthma, and only palliative treatments have been developed to help these people, no definite cures. However, if we can isolate and identify asthmatic agents in the biosphere, both biotic and abiotic, then we could potentially generate more targeted treatments for asthma and in doing so be a step closer to finding a cure. One potential asthmatic agent, Pichia-Kudryavtsevi yeast, has been recently researched in vivo studies. For example, one study found the same yeast present in the feces of infants in Ecuador, and those same infants were then more prone to developing asthma by the age of 5. But as we know, correlation does not always mean causation, which is why in this study I decided to test the direct effects of Pichia-Kudryavtsevi yeast secretions on A549 alveolar epithelial cells as to determine whether these secretions triggered any type of asthmatic behavior in the cells. To do this, I grew the cells in 6 well plates and inserted a 0.2 micrometer membrane per well, on top of which I grew the Pichia-Kudryavtsevi yeast for the desired treatment duration, which were either 4 hours, 0 hours, 6 hours, 12 hours, 24 hours, or 48 hours. After the treatments, I tested the infected samples via a trypan blue assay for cell proliferation, an ELISA IL-6 assay to test for cytokine hypersecretion, a tunnel assay to determine the amount of apoptotic cells, and a PIS staining assay to quantify mucin production. After statistical analysis, results showed that the yeast secretions did have significant effects on the asthmatic behavior of the cells. In a majority of the samples, the yeast secretions increased apoptosis, caused IL-6 hypersecretion, led to mucin overproduction, and significantly decreased the viability of the cells. Thus, I was able to arrive at the conclusion that Pichia-Kudryavtsevi does in fact have characteristics of an asthmatic agent. This discovery has many implications in the medical field as well as many future applications that could greatly expand the scope of asthma research. The next step in the direction of this experiment would be to increase the complexity of the model organism being used in order to see the broader effects of the yeast secretions at the tissue or even organismal level. Furthermore, analyzing the contents of Pichia yeast secretions could be helpful in identifying the molecules responsible for this asthmatic behavior and thus bring us a step closer to finding a cure. Thank you for watching.